All right, so let's get going today. So today's topic is bioenergetics. So we're looking at how energy comes into a system and moves through it and ultimately out of the system. Three main topics we're going to talk about today, if we get to them all, will be enzymes, photosynthesis, and cell respiration. We may not get to all of those today. We'll just have to see how time goes. But we're going to focus today on some FRQs, some free response questions, because that tends to be a weakness of every student pretty much in AP Bio. We're going to start each of those topics with a free response question. So I have one about enzymes up on the screen now. I'd like for you to go ahead and read the FRQ, and then I'm going to ask you to do some things with that FRQ here in just a minute. I'm also going to ask you to jump back and forth between the Zoom screen and the Pear Deck screen. And the reason that I'm going to do that is I can write on the PowerPoint screen and you can see it. I can't do that with the Pear Deck screen. So just be listening. I'll tell you to be either on the Zoom screen or on the Pear Deck screen. Right this second, it doesn't matter which one you're on. They both should have the same thing on the screen. We want you to read the FRQ and look at that data table. Take about a minute and do that. Oops, let's try that again. All right, so really what you need to address this question is the equation that you see at the top of your screen. And I'd like for you guys to go back to the Zoom screen with me for a second as I'm writing a few things on the screen for you. So this equation is really important. Let's see if I can get my pen to work here. Nope. Give me just a second to get my pen going. So that equation. So there's this substance that's in pretty much all plants called catechol. It reacts with oxygen with the help of an enzyme, polyphenol oxidase, and it makes this benzoquinone chemical. This chemical builds up in apples especially, and it's this chemical that makes them turn brown. So catechol converted by the enzyme polyphenol oxidase to this substance, and the buildup of that substance makes the apples turn brown. So some bright student noticed that if you altered the pH that the lemons were, that the apples rather were in, by using lemon juice, that you could slow down how fast they turn brown. So if we go down to the bottom of the screen here, on the axes provided, construct an appropriately labeled bar graph to analyze the effect of pH on polyphenol oxidase activity. So first of all, always pay attention to the verbs in the screen, or in the question rather. We call these command terms. These tell you how to get the point. So we're making a graph Also, make sure that you notice it says on the axes provided. That's going to be on the AP exam for you. Appropriately labeled, and it's specifically asking for a bar graph. And we're looking for the effect of pH on polyphenol oxidase activity. So let's go to the next slide. So this would be what your graph looks like. Someone unmute and tell me, what variable do you typically put on the x-axis when you're making a scientific graph? What variable goes on the x-axis? Somebody unmute and tell me. Go ahead.
Independent variable.
Yeah, the independent variable. And Mr. Patel, in this case, what would the independent variable be?
The pH.
The pH. Because remember, the independent variable is the thing that you're intentionally manipulating. Sometimes we call the independent variable the manipulated variable. So we want to graph that on the x-axis. So let's label that pH. And notice our pHs run from 1 up to, let me let in these folks trying to get in real quick. So our pHs run from 1 to 11. So I'm going to skip about every, say, 2 lines, and I'm going to make each one of those be 1 pH unit. Something like so. So this is going to be 1, 2, 3, 4, and so on. And you got to make sure you label. All right, and what do we graph on the y-axis typically? Someone unmute and tell me that. What are we going to put on the y-axis?
The dependent variable.
The dependent variable. And remember, the dependent variable is sometimes called the responding variable. It's the thing that changes because you changed pH. And in this case, our dependent variable is this. We're looking at the absorbance. So we're essentially, what we're doing here is we're shining a light through a tube with this chemical in it, and we're measuring how much of the light that started on one side is absorbed before it makes it to a sensor on the other side. And I hope what you understand here is that the browner that tube gets, the more light it's going to absorb. And remember that the brownness is showing us the buildup of that benzoquinone stuff. So we're going to put absorbance over here. I'm just going to label it like so. If you have units, it's a good idea to do that. We don't really have units here because we're just looking at a change in absorbance. Really, that's probably the percent absorbance. And the pH is just in pH units. So there's really not any units to put down there on the bottom. Now this says to do a bar graph. Specifically says a bar graph. So let's look at what my Y units are. So it looks like I go from about 0.06 all the way up to 0.32. Let's see, what would be an appropriate unit here? Let's make each one of these lines be 0.05. Actually, that's going to make them awfully small. Let's see if we can make a little bit better here. Let's see, what would be good? 0.02. Yeah, I'm gonna go with 0.05 just for the sake of time. As long as you can show the pattern, you are okay. All right, so at pH 1, our absorbance is 0.06. So somewhere like that. At pH 3, 0.09. pH 5.23. pH 7.83. So we need to go up further. I didn't see the 0.83 there. All right, so we're at 0.83 at 7, so let's go way up here. 0.83, somewhere in this neighborhood. Whoa, that's a really curvy line, guys. You definitely would want to use a straight edge here. You don't want a curvy bar graph. 9 at 0.32. So somewhere in there. And 11 at 0.10. So somewhere in there. And other than the curviness of my bars, that's a pretty decent bar graph. Independent variable on the x-axis, dependent variable on the y-axis, label and number your axes, put units on there if you have them, and then just plot your points as a bar graph. Make sure you do a bar. So hopefully that's pretty straightforward. Now let's go to the next part of the question. By the way, here's the grading rubric. This is how you would be graded on the part A. So notice you could have earned 3 points on part A. 1 point for labeling your axis correctly, pH on the X, change in A99 after incubation with enzyme on the Y. You correctly scaled it, so your lines made sense as far as what the lines represented. And then that you correctly made a bar graph. And notice it's ensuring that pH is the independent variable, and that should be on the x-axis. So that's how you get those 3 points. Now, part B, based on the experimental results, estimate the optimal pH of the enzyme. Explain your estimate by connecting the experimental results to the structure and function of enzymes. So first of all, we're estimating. So somebody unmute and tell me the optimal pH for this enzyme.
7.
7. And the reason it's 7 is because that's where the absorbance is the highest, which means that's where the tube becomes the brownest. That's where the most of the catechol has been converted into that benzoquinone chemical. It's the most effective. So the first thing you're going to say is that the optimal pH is around 7. Next, explain your estimate by connecting the experimental results to the structure and function of enzymes. So what I would say here is that each enzyme has a pH that it works best at. If the pH becomes, if the pH goes down and becomes more acidic, or if the pH goes up and becomes more basic, let's say your optimal pH is right in here. What happens when you move away from that optimal pH in either direction, too basic or too acidic? What happens in terms of the structure and function of a protein? Somebody unmute and tell me.
Denaturation.
Yeah, the protein denatures. Now that don't just say denatures though, because that's talking about the structure. So you would say the protein denatures, or you could say it changes shape. That's what denaturation means. And therefore it ceases to function, or it functions more slowly. So really, to get the points on this, let's look at the rubric on this one. So here's part B. So you got 1 point for estimating that 7 was the optimal pH. You earned another point for talking about evidence. The reaction has the highest absorbance at that pH, something like so. And then you got a point for justifying, talking about the fact that when you move the pH away from the optimal pH, the enzyme denatures, it changes shape or structure, and therefore it reduces its activity. So you could have earned 3 points on part B as well. Since I've already got it pulled up, C says propose an appropriate control treatment for the experiment and describe how the control treatment would increase the validity of the results. So remember that we already had, let's go back to our table for a second. So if we go back to our table, we had all these pHs from 1 up to 11, odd numbers, and then we measured absorbance. So as far as the control goes, some of you might say, well, the control ought to be pH 7. But the thing is, we already have pH 7. That's part of our experimental protocol. That's part of our independent variable. So the control here could have been any of these things you see in the list. It could have been that you ran the experiment without the enzyme. And the description, why that's a good control, is that shows you that the enzyme is necessary. You could have ran it with an enzyme that you know was denatured, that didn't work to begin with. And again, this shows you that you've got to have a working enzyme for the reaction to happen. You could have run it under anaerobic conditions, because if you remember, that formula for the reaction required oxygen. So that could show that oxygen is required to make this happen. Finally, you could have run it without the catechol compound. Remember, the catechol compound is the original substrate. So by running a reaction without that, you could confirm that it is the original substrate. It's necessary for this reaction to happen. So 2 points possible, 1 for giving an appropriate control, and 2 for describing why it's an appropriate control. All right, last. Genetic engineering can be used to disrupt specific genes in the genome of an organism. Predict how the browning of apple slices would be affected in a strain of apples that was genetically engineered to lack functional copies of the gene encoding phenol oxidase. Well, I'm hoping this is an easy part of the question for you. We've already got the rubric up here, 2 points. Remember, this is the enzyme that causes catechol to be converted to the benzoquinone, which turns the apple brown. So if the enzyme doesn't work, that should mean the benzoquinone is never formed, and therefore the apple doesn't turn brown. So your prediction, apples won't turn brown, or they'll do so more slowly. Either one of those is okay, one of those points. And then 2, the justification, browning requires an enzyme, or something like you probably could go benzoquinone causes the apples to turn brown. Without the enzyme, benzoquinone won't be formed. At least it won't be formed as quickly. So something like that works. All right, so questions on this FRQ? What I'm hoping that you see is, without knowing a lot of biology, most of you could have answered several parts of this question. Pay attention to what the question's asking. Pay attention to these verbs. Justify is a really, really important verb in AP biology. Talk about the evidence. So make a prediction and then use the evidence to explain why you made that prediction. Back up your prediction with evidence. That's what we mean by justify. So I've got a few slides here about enzymes that I want us to look at. Just a quick review of enzymes other than what we did on the FRQ. So here's an enzyme. Remember, enzymes are proteins with a very specific 3-dimensional shape. There's one section of that enzyme called the active site. It has a very specific shape and a very specific charge. And the substrates of the reaction, they have to have a shape and a charge that are complementary to that active site. They have to fit in it sort of like a key fits into a lock. And when they do fit together, that's called the enzyme substrate complex. And that's when the enzyme actually starts to work. It's when it catalyzes reactions. Remember, that's really what enzymes are. They're catalysts. They make reactions happen by using less activation energy. So they make reactions that would normally require a lot of input energy to happen without very much energy. In this case, this enzyme is what we call a catabolic enzyme. Anybody able to unmute and tell me what catabolic means? Anybody out there? Catabolic, what does it mean? What's happening in this reaction to the substrate? Come on, don't be shy. Somebody tell me what's happening to the substrate in this particular reaction.
It's breaking the bonds.
Broken down, exactly. Catabolic means breakdown. And somebody else tell me what's the opposite of catabolic?
Anabolic.
Anabolic. So enzymes are usually one or the other. There are some enzymes that are catabolic that break things down. There are other enzymes that catalyze reactions that put things together that are anabolic. Either way, when the reaction's over, we now have these products, in this case, glucose and fructose. And notice the enzyme, it doesn't look any different than it did at the beginning. The enzyme is unchanged by the reaction, and it can be used over and over again. So in an enzyme catalyzed reaction, the substrate's eventually going to get used up. It's going to run out. But the enzyme doesn't run out. It can be used over and over and over and over again to catalyze the reaction that produces the products from the initial substrates. This reaction is showing you that when the substrate fits into that active site, that the enzyme itself actually changes shape just a little bit. And that change in shape, a tiny change in shape, is called an induced fit. And when it changes shape, that causes the enzyme to actually work. So in this case, probably what's happening once this substrate binds in, this protein is probably bending out a little bit, like so. And when it does, it torques the bond between these two pieces, and eventually it breaks them apart. So this is a very small change in shape. This is not like denaturation. This is a small change in shape, an induced fit, that actually causes the enzyme to do its job, causes it to work. Here we just see that each enzyme has a temperature that it works best at, an optimum temperature. And for most enzymes in the human body, that temperature is 37 degrees C or 98.6 degrees Fahrenheit, body temperature. If you go higher than that, what starts to happen? If you took a human enzyme, say in a test tube, and you raised it above 37, what's going to happen to that enzyme?
It starts to denature.
It starts to denature, and therefore the rate of its reactions are going to drop very quickly. Now, on the other hand, if you go this way, cold doesn't denature things. But remember that reactions happen when molecules bump into each other. And if you get cold, why would that happen more slowly? Why would reactions happen more slowly in the cold? Somebody help me out. Why do reactions tend to happen more slowly as it gets colder? What is heat really a measure of? Or temperature? What is temperature actually measuring? It's measuring how fast molecules are moving. Think of a thermometer like a radar gun for molecules. And when they're moving fast, the temperature's high. When they're moving slow, the temperature's low. So as the temperature decreases, the molecules in the container or in the body are moving slower, and therefore they don't bump into each other as often, and therefore they don't react as much. So that's why as you go colder, the reaction rate drops. Now, on the other hand, each reaction also has an optimum pH. a pH that it works best at. For most human enzymes, that's 7 or around 7. And if you move either this way, where it becomes more acidic, or you move this way, where it becomes more basic, the reaction rate drops very quickly as you move away from 7. And that's because the enzymes are denaturing. Acids and bases can cause the enzymes to essentially fall apart, to change shape, so they don't work. Now, there are a few enzymes in the body that have acidic pHs, acidic optimal pHs, like the ones in your stomach. The pH in your stomach is somewhere around 2. So you have to have enzymes that can work effectively at that pH. All right, this is reminding us that enzymes also can have what are called inhibitors. In this case, we have what's called a competitive inhibitor. This is a substance, in this case, this dark blue substance, that actually binds to the active site of the enzyme and prevents the substrate from binding there. So if the inhibitor's there, the substrate can't be. And since the substrate can't fit into the active site, the enzyme doesn't work. The reaction doesn't happen. Now, there are also another kind of inhibitor called the non-competitive inhibitor. These are non-competitive because they're not binding to the active site. It's still open. They bind to some other site on the enzyme. Sometimes it's like an allosteric or a feedback site. And when they bind there, that actually causes this active site to change shape a little bit. And when it changes shape, the substrate still can't get in there. Regardless, doesn't matter if it's a competitive or a non-competitive inhibitor. Inhibitor means to stop. So inhibitors are substances that attach to an enzyme and cause it not to work. If the substance attaches to the active site, it's a competitive inhibitor. If it attaches somewhere else, it's non-competitive. All right, we also want to talk about something called feedback inhibition. And this is actually usually a good thing. It's an example of negative feedback. So let's say this end product is something that your body needs. And all these steps, all these enzymes that you see in this step are what we call a metabolic pathway. So let's say that, let's just say that this is some kind of hormone that your body needs. And all these reactions and all these steps make this hormone. You don't need an unlimited amount of that hormone. You only need a tiny amount probably. So it doesn't make sense for your body to continually make it 24/7. It makes as much as it needs, and then it stops. And this process of feedback inhibition allows your body to regulate when a metabolic pathway is on and when it's off. So notice in the process of making this hormone, we start with a reactant. In this case, it's called reactant A. And enzyme number 1 catalyzes a reaction. These arrows represent reactions, chemical reactions, that convert substance A to some new chemical, chemical B. Well, a second enzyme comes in and catalyzes a reaction that converts substance B to C. A third enzyme comes in and converts substance C to D. A fourth enzyme comes in and converts D to E. And finally, a fifth enzyme comes in and converts E to what we're trying to make. So each one of these steps is crucial for making this end product. And each one of these substances that are made between the initial reactant and the final product, they're called intermediates. Hope you remember that inter means between. So they're things that are made between the first reactant and the final product. Point being, though, we're making this hormone. As the concentration of this hormone starts to build up in your cell, some of it bumps into usually the first enzyme in the pathway, E1. And it turns out some enzymes have 2 grooves on them. The active site, which you've already talked about, and another site called the allosteric, or sometimes it's called the feedback site. And it turns out what fits into that groove is the final product of the pathway. So if some of this end product bumps into that enzyme and fits in that groove, that causes the active side of this enzyme to change. That means this enzyme doesn't work anymore. Well, that means that B is not made anymore, which means that E is not working anymore, which means that this isn't made, which means this isn't working anymore, which means this isn't working anymore. That isn't working anymore. That's not made. That isn't working anymore. And therefore, your body doesn't make any more of that. So by shutting down the first or one of the very early enzymes in the pathway, the whole metabolic pathway is shut down. But it's not shut down permanently. It's shut down until the concentration of F starts to get used up. Concentration drops. And when the concentration gets low enough, the piece of F that was sticking here lets go. And it gets used up. And when that piece gets used up and it's not attached to this allosteric side anymore, the active site rather goes back to normal. Now enzyme one starts to work. It starts to convert A to B. Now enzyme 2 can work and it can convert B to C. Ultimately, more of this starts to get made. So feedback inhibition is a way in which the end product of a pathway deactivates the whole pathway when its concentration gets high. But when its concentration gets low, the pathway starts back up and it begins to create more and more of this. So this is your body being conservative. Your body making what it needs, and then when it's got enough of it, pausing, using that up, and then when it starts to get low, turn the pathway back on and make more. That's feedback inhibition. All right, any questions so far? All right, well, let's look at another FRQ. And I think instead of working from the Pear Deck screen, I think we're just going to work from here for the sake of time. So I want you guys to take a minute and just read what you see on the screen there. So read what you see on the screen. Look at the graphs 1 and 2 up at the top, and also pay attention to the stuff there in the middle. So this is really, we're talking about what are called absorption spectra. And this essentially shows us how much light is absorbed by certain pigments. And we're breaking it down by the color or the wavelength of light. So it's telling us that one of the pigments is chlorophyll A, main kind of chlorophyll, kind in green plants. And the other one is bacteriorhodopsin, which is this purple-like pigment found in some photosynthetic bacteria. And the first thing, notice part A, identify the pigment used to generate the absorption spectrum in each of the graphs. Explain and justify your answer. So notice you've got 3 bold words. You got to make sure you do all 3 of those. So let's go up to the to graph. Let's just look at graph 1 first. Somebody unmute and tell me, do you think that's bacteriorhodopsin or chlorophyll A? Graph 1. Bacteriorhodopsin or chlorophyll A? Someone unmute and tell me what you think.
Chlorophyll A.
All right. Why do you think chlorophyll A?
Since it was talking about like green plants, I just assumed that it was higher over there in that range.
So let's think for a minute. Why are green plants green, guys? What makes green plants be green?
I think they reflect that green light.
Yeah, so they're reflecting green light. That means they're not doing what to green light?
Absorbing it.
They're not absorbing it. So if we look at graph 1, whatever this is in graph 1 is really good at absorbing somewhere between 500 and say 600, all right? And if you look at that band, that's green. So whatever's going on in here, these guys are absorbing green. That's telling me that's not chlorophyll. But if we go over to graph 2, Notice that between 500 and 600, they're not absorbing that at all. When they're not absorbing, they're reflecting. So graph 2 is showing us something that's reflecting green light, a green plant. That's chlorophyll A. Would help if I spelled it right. Now, let's think about what's going on with the other one here. So this one's absorbing green and yellows, but it's reflecting the 400 band, which is bluish. It also seems to be reflecting around 700, which is reddish. So if you've got red and blue together, what do you see?
Purple.
Purple. So this is bacteriorhodopsin. So again, identify the pigment. So you should say graph 1 represents bacteriorhodopsin. Graph 2 represents chlorophyll A. That answers just the first part of that question. Identify the pigment. Now explain and justify your answer. Well, let's go to bacteriorhodopsin. And we could say something like this. Bacteriorhodopsin, or the pigment in graph 1, is reflecting bluish light, 380 to 475, something like that. It's also reflecting reddish light, 620 to 750, and therefore it appears purple. On the other hand, you could say in graph 2 represents chlorophyll A because it seems to be absorbing blue light and red light very well, but it's reflecting or not absorbing light in the 495 to 570 range, which is green, which means it's reflecting green and would appear to be green to the human eye. So that's the explain and justify part. So if we look at the rubric on that, again, you got 1 point for identifying both pigments, and you needed to say graph 1 is, graph 2 is. And then the whole explain thing, you could get 2 points if you explained it and justified your answer. 3 total points on part A. So now if we go back to part B, in an experiment, identical organisms containing the pigment from graph 2 as the predominant light capturing pigment are separated into 3 groups. Remember, graph 2 was chlorophyll. So this is a green plant or something like it, maybe an algae. The organisms are grouped, in each group are illuminated with light of a single wavelength. So one color, each group gets a certain color. One group gets 650, one group gets 550, and one group gets 430 rather. The 3 light sources are of equal intensity, and all organisms are illuminated for equal lengths of time. Predict the relative rate of photosynthesis in each of these 3 groups. Justify your predictions. So remember, 650, 550, and 430. Let's go back to the table. So 650 is going to be red light. So this one's red. Let's go back to the table. 550 is right in here. So 550 is green light. And 430, right in here, blue light. Now let's go back and look at our absorption spectrum real quick. So I'm hoping that you notice that chlorophyll is best at absorbing blue. It's got maximum absorbance there. That's that 430. It's second best at absorbing red. That's the, was it 650? 650. And in here in the 550, it's basically doing nothing. So as far as the question goes, I'm hoping that you see your prediction would be something like this. Plants, well, first of all, you guys tell me, which one should do the most photosynthesis? Which one should do photosynthesis the fastest? 650, 550, 430. Help me out.
4:30.
430. And you should say something like this. I predict that the plants exposed to 430 nanometer light will do photosynthesis the fastest because chlorophyll A is best at absorbing light of that wavelength. And therefore plants exposed to that light have more energy to do more photosynthesis. The plant that would do photosynthesis second fastest would be the plants exposed to red light, 650 nanometer light, because that's the second best wavelength of light that plants absorb. And therefore plants that are exposed to that get the second most energy and can do the second fastest photosynthesis. Finally, the plants in the 550 nanometer group, the green light group, essentially they reflect all that light. They absorb almost no light, and therefore they have no energy for doing photosynthesis. So really we kind of did all three of those things in the same sentences. We predicted fastest to slowest, and then we justified by talking about what forms of light were absorbed the best. Blue the best, red the second best, green pretty much none at all. All right, and then finally, part C, bacterial rhodopsin has been found in aquatic organisms whose ancestors existed before the ancestors of plants evolved in the same environment. So we've got these bacteria, probably maybe in a pond kind of area or maybe the ocean. They evolved first. So they're the first photosynthesizers. They've got this purple pigment, bacterial rhodopsin. Propose a possible evolutionary history of plants that could have resulted in a predominant photosynthetic system that uses only some of the colors of the visible light spectrum. All right, I'm going to go ahead and look at the rubric on this one. So remember, plants don't use all the colors of light. Some of you might think, well, that doesn't make any sense. If all that light's shining on them, why don't they use it all? So one answer could be this. Absorbing all the wavelengths of light could be too much energy. A lot of times when energy is absorbed, like let's say you had on a black t-shirt out in the summer, you get really, really hot, really, really fast. Well, you have legs, so you can get up and get in the shade, or you can go in the house. Plants can't move. So if plants were black and they absorbed all wavelengths of light, they would very quickly overheat, probably die. So that might be one thing, one proposal for why they don't use all the wavelengths of light. It would be too much energy. And that natural selection favored organisms that only, that used pigments that only absorb certain kinds of light so that they didn't get too hot. Now, another possible answer, let's say that instead they evolved in the same environment as the bacteria using bacterial rhodopsin. That means that the reds and the blues, the greens are being absorbed by these guys. So these guys are absorbing greens and yellows. Let's say they live in the top couple of layers of the water. Whatever light gets down to the plants a little bit deeper, the greens and the yellows have already been used. And what makes it down there are the reds and the blues. So it could be that they evolved that way because green light was already being used up. It wasn't available. It was being used by other things in the water. And again, natural selection favored organisms that could use the kinds of light that were available, which happened to be the reds and the blues. So point being, part C, there were a lot of different ways to answer it, but you had to have a good proposal and then an explanation for why that could have happened the way that it did. All right, I intended on spending some time going through the process of photosynthesis. I don't think I really have time to do much there. One thing you should know though is the equation, the overall balanced equation for photosynthesis. You need sunlight, you need carbon dioxide and water. And you know that the carbon dioxide comes in through the stomates or the stomata, the holes on the bottoms of the leaves. The water comes in through the roots, and the plants use the energy from the sun, and essentially they put together carbon dioxide and water to make sugar, carbohydrates. Oftentimes we draw that as glucose, C6H12O6, but they can make other sugars as well. And then they give off oxygen gas, which is O2. If we balance this guy, we need 6, 6, and 6, and 1 there to balance it out. Another important thing to realize is that plants do need oxygen gas. Somebody tell me what plants need oxygen gas for. What do plants need O2 for? Somebody help me out. What do plants need O2 for?
Cellular respiration.
Cellular respiration. A lot of people think plants do photosynthesis, animals do cell respiration. Well, that's sort of true, but what the best answer is that plants do both. Plants make the carbohydrate through photosynthesis, and they store it. And then when they need to use it, they break down the carbohydrate and get the energy out through cell respiration, which means they do have to have some oxygen. Now, it turns out they make more than they use. So they're releasing extra oxygen out into the environment, but they're using some of that oxygen as well. Plants do both. They do photosynthesis and cell respiration. Every organism on Earth that we know of does some kind of cell respiration. That's important to know. Whether we're talking about bacteria, fungi, plant, animal, or anything in between, every living thing does cell respiration. So photosynthesis, at least in eukaryotes, happens in the chloroplast. Most of those are in the leaves. And the pigments are actually located in the membranes of the thylakoids, these little sacs that stack up inside the chloroplast. The first part of photosynthesis is sometimes called the light-dependent reactions. And essentially what happens in those steps, the plants absorb light. That absorbed light energy causes electrons to move. It is used to generate a proton gradient inside the thylakoid. And then those protons move out through ATP synthase and provide the energy to make ATP. Also made during this process is another energy storage molecule called NADPH. And this is also where O2 gets made. So the light-dependent reactions make 3 important substances, ATP and NADPH, both forms of stored energy, and then also O2. Now, the downside of these 2 kinds of energy is that they're both really reactive molecules, and they're both really hard for a plant to store or transport down to the roots or to the stems. So they need something that's more storable and more transportable. And that's when part 2 of photosynthesis, called the Calvin cycle, or the light-independent reactions, happens. The Calvin cycle takes in carbon dioxide from the stomata, reacts it with other molecules already found inside the plant, in this case, ribulose bisphosphate. And then it adds to that the ATP, the energy from the ATP made in the light-dependent reactions, and the electrons and energy from the NADPH, also from the light reactions, to make a molecule called glyceraldehyde 3-phosphate, or G3P. This molecule is essentially half of a glucose. So plants can take 2 of these and put them together and make glucose. Or they can take other ratios of glyceraldehyde 3-phosphates to make other sugars or other organic molecules that the plant needs. Point being, in the Calvin cycle, the plant takes in carbon dioxide and uses the energy storage molecules, ATP and NADPH, from the light-dependent reactions to make carbohydrates. Carbohydrates are ways to store energy that can be transported. That's why they're so crucial to the plant. They're storable and transportable, ATP and NADPH, not so much. All right, so we're going to end today by looking at this last FRQ. So take just a minute and read the whole thing, and then we'll talk you through it, and we'll call it a day. All right, so really this is a question about the carbon cycle. And there's two main processes in the carbon cycle. One of them takes carbon dioxide and uses that to make organic molecules. So notice our first question, identify the key metabolic process for step 1 and the key metabolic process for step 2. So somebody unmute and tell me what step 1 is. What does step 1 represent?
Photosynthesis.
Photosynthesis. So you should say step 1 represents photosynthesis. Period. That's a point. All right. Another process takes organic molecules and converts those into carbon dioxide.
Cellular respiration.
That's cellular respiration. So you should say step 2 represents cellular respiration. Without knowing a whole lot of biology, a lot of you could have answered those two things. So you've now done this first part. Next, briefly explain how each process promotes the movement of carbon through the cycle. Also, for each process, your explanation should focus on the role of energy in the movement of carbon. So I would say something like, for photosynthesis, for step 1, plants absorb sunlight energy and use that energy to react carbon dioxide from the air, to take in carbon dioxide from the air, react it with water to make sugar, which is a form of stored and transportable energy for plants. So that sort of does the explain part for 1. And then for 2, you could say during cell respiration, step 2, organisms break down organic molecules in order to get the energy out of those molecules. They use that energy to make ATP, which fuels essentially all the reactions in the body. When the organic molecules are broken all the way down, the leftovers, essentially the waste, are released into the environment as carbon dioxide. So essentially CO2 is what's left over and unusable from the organic molecules. And that's really what they wanted you to talk about for the explain part of step 2. And then finally, for B, identify an organism that carries out both processes. Somebody help me out. What's an answer for B?
A flower.
So, I didn't hear you. Say again.
Flower.
Yeah. So you could say, I wouldn't say a flower, I would say a plant, but you could say any plant, you could say any algae, you could even say some photosynthetic bacteria. Anything that does photosynthesis also does cell respiration, because everything does cell respiration. So you could name any specific plant, you could just say a plant, you could say an algae, you could say a photosynthetic bacteria. That's really all there is to it. So again, this question, really simple. A lot of them are this simple. You just have to read the question and make sure that you address those key command words, those bold words in each question. All right, before you guys go, I'm going to go back to the Pear Deck screen for just a second. Even though we didn't really use Pear Deck today, you want to make sure that you sign in. so that you get credit for being here today. And you'll get a copy of the presentation. So if you did not sign into Pear Deck, make sure you go to joinpd.com, joinpd.com, and enter the code that you see in the upper right-hand side of my screen, AALGRQ. And once I end the session today, you guys will get a copy of my presentation. And that way, some of you I know are using that as proof that you were here today. So make sure, again, you go to joinpd.com and you enter code AALGRQ. All right, any questions I can answer for anybody today?